अस्सलाम वालेकुम, मैं मोहम्मद उमर वजदान फ्रॉम द डिपार्टमेंट ऑफ बायोटेक्नोलॉजी 3rd सेमेस्टर। मैं सबसे पहले फ्रंटियर वॉइस का शुक्रिया अदा करना चाहूँगा क्योंकि उन्होंने हमारे लिए ऐसा एक साइंटिफिक प्लेटफॉर्म अरेंज किया है जिसमें आज साइंटिफिक पोस्टर कंपटीशन है। हमारे ज़िम्मे मतलब जो भी आइडियाज़ है, प्रोजेक्ट्स है, रिसर्चेज है, तो हमने उन्हें एक पोस्टर के फॉर्म में पोर्ट्रेयल उसका किया है और उन्हीं पोस्टर को हम मुख्तलिफ लोगों को, प्रोफेसर्स को, रिसर्चर्स को दिखाएंगे कि हम अपना आइडिया, हमारे अपने इनोवेटिव सोच और जितने भी हमारे आइडियाज़ हैं वो हम कन्वे करें इन अ प्रॉपर एंड फंडामेंटल वे। तो फिर से मैं फ्रंटियर वॉइस का शुक्रिया अदा करूँगा कि ऐसी प्लेटफॉर्म उन्होंने हमारे लिए अरेंज किया है एंड आई एम वेरी हैप्पी अबाउट इट।
अस्सलाम वालेकुम, मेरा नाम जवेरिया हक नवाज़ है और मैं डिपार्टमेंट ऑफ बायोटेक्नोलॉजी से हूँ और मैंने आज साइंटिफिक पोस्टर कंपटीशन में पार्ट लिया है। और सबसे पहले तो मैं फ्रंटियर वॉइस का शुक्रिया अदा करना चाहती हूँ कि उन्होंने इस तरीके से टैलेंट हंट अरेंज किया। ये था कि हमारे रिसर्चेस के बारे में और तहकीक के बारे में एक प्लेटफॉर्म ये हमें दें और जहाँ पर हम लोग दूसरों को बताएँ और दूसरों को अपने ये रिसर्चेस और तहकीक के बारे में बताएँ। और इस तरीके से हम हुए हैं, यानी हमने पार्टिसिपेट किया है तो दूसरे स्टूडेंट्स को भी मौका मिलेगा कि वो भी आएंगे और पार्टिसिपेट करेंगे।
बिस्मिल्लाहिर्रहमानिर्रहीम। थैंक यू वेरी मच। आज ये फ्रंटियर वॉइस की तरफ से स्टूडेंट के लिए एक अपॉर्चुनिटी है जिसमें ये लोग साइंटिफिक पोस्टर जो है वो उसकी एग्जीबिशन है। ये एग्जीबिशन बेसिकली यूनिवर्सिटी में स्टूडेंट आते हैं, क्लासरूम्स, क्लास और घर, लेकिन ये जो इस तरह की एक्टिविटी है उनकी फ्यूचर ग्रूमिंग के लिए बहुत ज़्यादा ज़रूरी है। और इसमें जो स्टूडेंट है उनको रिसर्च के हवाले से पता चलता है कि आया रिसर्च कैसे होती है, उसका फ़ायदा क्या होगा, उसकी कम्युनिटी के लिए क्या फ़ायदा होगा। तो इस हवाले से जो है हमारे स्टूडेंट को ये मौका ये फ्रंटियर वॉइस की तरफ से मिल रहा है और हम इनके शुक्रगुजार हैं कि इस तरह के इवेंट ये ऑर्गेनाइज करते रहें। थैंक यू वेरी मच।
My PhD from Department of Microbiology, Abdul Wali University, Mardan. The title of this presentation is the virulence potential of the polysaccharide dependent biofilm forming Staphylococcus epidermidis. Staphylococcus epidermidis is an opportunistic bacterial pathogen. Basically, it is a normal flora, but in certain special situation, it causes infection. Infection is due to one of its main virulent determinant called the biofilm. So far, the composition of the biofilm is related. So far, it is concluded that this biofilm is due to the polysaccharide secreted by the bacteria. But in our initial screening, our initial genetic screening, we checked that the polysaccharide encoding genes are very low in this bacteria. So we speculated that the Staphylococcus epidermidis may not be a pathogenic one. It can be a potential contaminant because it is a skin normal flora. It can easily contaminate the samples. So for that, we checked that either now if the corresponding biofilm forming Staphylococcus epidermidis is due to a polysaccharide, then it may be a contaminant. So we screened the potential of the polysaccharide dependent biofilm forming Staphylococcus epidermidis and the polysaccharide independent biofilm forming Staphylococcus epidermidis. We screened its virulence potential. To answer this question, the differential proteomic approach was followed, in which the question was answered by this methodology, in which initially the samples were taken from bacteremia patients and then they were processed for their genotypic and physiological analysis was performed. After genotypic analysis, then they were transferred to the proteomic analysis, in which the proteins were extracted from the total cell lysate, followed by the mass spectrometric analysis. Expressed proteins were checked.
Nice research has been conducted, but in case it has not been coated, biofilm has not been supposed to be over the bacteria, what could be the consequences of that particular bacteria then?
If it is not able to produce a biofilm?
Yeah.
Biofilm is basically the initial stage in this bacteria. It is the actual virulence determinant. If the bacteria is not able to produce the biofilm, then it will not be able to initiate the infection. Actually, when we initially see that from where the infection starts, it starts with the insertion of a cannula. When a cannula is inserted, it takes the nerve flora from the skin toward the vein. Then after that, 24 to 48 hours, after 48 hours, it then first produces a biofilm. Then from the biofilm, the bacteria are in static condition, but now from the biofilm, when the biofilm is matured, then the bacterial dissemination starts, systemic dissemination, then it causes bacteremia,
Right.
the bacterial presence in the blood. So after its biofilm production, then it will initiate the infection. So biofilm is critical for the pathogenesis.
Yes.
If it is not present, the bacteria will not be a pathogenic one.
Right. Does it affects the immunity system?
Of course, biofilm is the pathogenic determinant, and its actual pathogenicity is the immune evasion.
Yes.
So bacteria which will produce a strong biofilm will be a strong pathogenic one because of the evasion of the immune system. Besides this, it also increases the antibiotic resistance.
Yes.
The antibiotic penetration is very low in the biofilm.
Thank you.
Summarize your work, like we can read all those things. You just tell us that you have produced 4 products and what how you produce.
So my
what are
product is
their
just
applications.
lipase-based detergents. First one
Okay,
is
you have
which I was extracted from lipase-producing bacteria, which was extracted from soil.
okay.
That lipases have compatibility with different detergents. I have used Surf Excel.
In case this bacteria is lying there in the soil, what are the places of that bacteria which is there? And in case you have controlled that bacteria there, again, what are the places there?
Sir, I don't understand. Please repeat.
If the bacteria is there, there are these plus points of the bacteria also there, nitrogen-fixing bacteria and the others.
Yes, yes.
If that bacteria is there in the soil, what could it do there? It could raise the plant growth there, or it could help there, it could make the
Fats degradation. Fats degradation. जो soil में different nutrients होते हैं, lipid base की उसकी degradation करता है. Use lipase for
this
that.
particular bacteria there.
Yes, yes.
In
For that substrate
in
degradation.
case you suppose remove that bacteria from there, from that environment, then what would be the negative point or the demerits of that bacteria there?
Sir, soil is an enriched environment which contains millions, billions of
Yes,
bacteria.
but you have specified one bacteria there,
Yes, sir.
which is this one. You have isolated that one.
Yes, sir.
From where you did isolated it?
From which side
From
specifically?
the sample, yes. You have
wetland.
isolated it there in this department?
No, I isolate it from wetlands different at Charsata, जो different rivers से उसकी soil sample से.
Then you would have collected from there.
Yes, sir.
Where you have isolated it. There are a bunch
In
of
the lab,
bacteria
sorry,
in the
in
lab.
microbiology lab, yes, sir.
Developed a biosynthetic symbiote. It means that there is a microcontroller in which a living slime mold called the Phasarum polycephalum is infused in it. So we have achieved some sort of artificial intelligence by which we can prevent many diseases and disorders. Like if I start from the problem statement, we know that we humans generally, we people generally diagnose a specific disease after the symptoms appear. So that is okay, but there are some specific biomarkers that doesn't show any symptoms, clinical symptoms. While so, later on, it develops into a dangerous health-taking disease. So, for such problems, for such symptomless danger diseases like, let's suppose I say, amelanotic melanoma, a cancer which only appears as pinkish spots.
Or you have performed or you have designed something in your lab as well practically.
Sir, for practical purposes, I have a software.
Yeah, that's what I mean. Like this is like seems like you have a design in your mind and you have designed this through like AI software, etc. And you have studied well. Right?
Yes, sir.
But have you done any practical work on this project?
Sir, due to the lack of financial resources, such integrated and computational work seems a little bit hard to perform.
This is a good idea, but like its implementation and its like generation is also important. Like what I'm interested, have you developed this in your lab? Any part of this project?
Mortality rates and the symbiote mortality rates. And I've of course taken references from some researchers as well and performed simulations in Pymol BioRender about this model but lab works, I doubt that there is some sort of
Hypothetical computational or simulation or like.
but we have done simulations.
Generally, a biotechnology student, we want this technology to be developed. Yes, definitely.
Because you know that, as you said, there should be its plus points should definitely should go to the society. And a common perception in the society is when a person is having a huge number of symptoms of any kind of disease.
Yes, sir.
Then our people goes to the hospitals and then they are properly investigated. When they are investigated, the cancer, God forbid, the cancer
Yes.
goes to the final stages.
Yes.
So it's a wonderful
This is
idea.
a good idea.
Very good idea.
You must work on it.
Yes, sir.
Airways, and today my presentation topic is anti-cancer drug prototype benzophenone sulfonide amide. Because this is a teamwork, me and Dr. Saira Parman and Dr. Zaida lead it. These compounds, first we check on leukemia, in which leukemia these compounds show best results. These synthetic compounds show best result. So I again check this compound on breast cancer. Here is a structure of this compound. In this compound there is a 3 benzene rings which show the lipophilicity. Aromatic rings show lipophilicity, and there is also a amine group is present which bind to metals and protein ions and enhance apoptosis. And also in this molecule sulfonamide is present. The sulfonamide group is a known anti-cancer, anti-diabetic and antioxidant agents. In case of breast cancer, we provide drugs to the cells, but in cells there are 3 transporters, MRP1, PGP and ABCG2. These transporter overexpress. These transporter overexpress, though that express level, the transporter gene expression increase, the transporter level also increase, and the drug which we provided, these drug also increase out of the cells, which does not retain under the cells. Here is a dinorubicin, which is a known anti-cancer drug. Here we provide the dinorubicin to the cell, to the patient, but because of the overexpression of these transporter, the dinorubicin doesn't retain under the cells.
And medicinal plant extracts, and check these medicinal plant extract against MDR pathogen, and checked against gastrointestinal pathogens, E. coli, Shigella and Salmonella typhi, and also check the oral pathogens to Staph aureus, Staph mutant, and against skin pathogens MDR, Staph aureus. And then I have checked the collected HPLC fraction and checked this fraction against these MDR pathogens, and then I have identified that specific compounds which are active against these compounds, and then I have designed for anti-MDR skincare cream and soap and lip balm and herbal toothpaste, herbal soap and herbal cream and herbal lip balm. And I have also compared the activities of the commercial toothpaste and cream, and also the herbal cream is this is commercial and this is herbal, that I have checked. The zone shows.
So have you checked the
Yes, I have checked the comparison between the commercial toothpaste and the herbal toothpaste that I have prepared from my medicinal plants that I have used. I have found that the commercial toothpaste have no activity, no inhibitions against the MDR oral pathogens, while the herbal toothpaste have activity and they can inhibit the growth of the Staph mutant and Staph aureus pathogen.
From what kind of plants you have extracted that toothpaste?
Figonia critica. These are
Common names and first of these plants.
Azraki.
So my research project is characterization of alpha amylase producing from extremophiles of Khyber Pakhtunkhwa. What is amylase? Amylase is one of the most important industrial enzyme, and it breaks the complex carbohydrate into simple sugars. Due to this property, it can be used in industries like detergent, textiles, bioethanol production. सर जो एप्लीकेशन मैंने परफॉर्म की एक्टिविटी की है वो बता दूँ? सर ये क्रूड एमाइलेज है.
there was the involvement of various students by making these posters, and a wonderful research has been conducted by all the students and the faculty members who was involved there, and the organizations who has arranged this event, the credit goes to them, to the TFV and to the Abdul Wali University, the Career Development Center. And such kind of events are needed to be encouraged in future also, because it could boost up the students' research level, and these could have the exposure to the community also. So this work can really help the university, and it is a kind of a joint venture between the TFV and the university, and these kind of activities can further enhance the collaborations between the other organizations as well as the universities.
To share my views, so really all the words what we have analyzed, those all are admirable, and the ideas the student have presented. So I was just checking next generation business idea competition, so they are really commercializable if these students are encouraged, they are provided the opportunities, so they can work more in their fields, and all the products can be commercialized. And I also thankful to the TFE and our University UA and CDC for arranging this wonderful workshop or poster competition. So thank you.
Respected deans, faculty members, students, and representative of the TFV, I welcome you all. And I think this is something very important, very close to my heart, and this is actually a real activities that we should do. And our graduates, they should be exposed to the market. The market should be here, so we should have the collaboration. The collaboration is very important. I am really thankful to both of the organization, TFV and Syntax Technologies both. The list which I shared by the director, Industry Development Center, that is a very good list, because it also got this design, it got the hospitality management as well, it has got the microbiology and those areas, so it's covered everything.
आखिर में मैं दोबारा फ्रंटियर वॉइस का शुक्रिया अदा करना चाहती हूँ कि उन्होंने हमें प्लेटफॉर्म दिया।